Hi, I'm Drew Anderson. I'm a research specialist here at the Basket Wildlife Research and Education Center and at the Moflux site. And right now we're standing on one of our Moflux vegetation plots where we track forest composition, tree mortality, and the production of leaf and woody litter so that we can better understand forest structure and how that changes both over time and in response to stress factors such as drought. This video is going to provide a brief introduction into the methods that we use to track the life of trees and the production of leaf and woody litter in the forest. We have 24 plots that are distributed along 5 transects, each starting near the Moflux tower and extending about 250 meters into the forest. And on each of those 800 square meter plots, we tag and identify all the individual trees, we identify their species, and measure diameter at breast height, or DBH for short. We use a special tape to measure DBH. So we'll wrap this around the outer surface of the tree trunk at breast height, and we can read diameter right off the tape. This tree, for instance, is 27 centimeters in diameter, and this allows us to follow the life cycle of these trees. So every month, we'll go and conduct a mortality assessment and look for new mortality, and then every 2 to 3 years, we'll go and re-measure the diameter of all of our stems and look for saplings that have grown up into what we consider to be trees, and then we add those to our inventory. On each of the 24 plots, we make 2 litter measurements, and leaf litter collects in tubs like this one right here. Once a month, we'll go out and collect the litter from those tubs and return it to the lab for analysis. And with these data, we know how much leaf litter is being produced in the forest, which is both an important source of soil carbon and can tell us about how the health of the tree canopy might be changing over time. We have found relationships between drought stress and mortality that usually follow in the subsequent year. After the major drought of 2012, we found extremely high oak mortality, especially for black oak, for which we lost about 25% of the live stems. We found strong evidence that the mortality was related to a set of interacting factors. So generally speaking, the drought stress weakened the trees to the point of them being even more susceptible to attack from pests and pathogens. In 2012, when our oaks were stressed, the weakest stems were overrun by a pathogen called Biscogniaxia, which is formerly known as hypoxylon, and it rots the sapwood of the trees. We found that trees that died in 2013 were heavily infected and historically had lower growth rates, and therefore were more likely to succumb to the pathogen. Hopefully you now know how we track the life of our trees in this forest, but before we go, I'm going to summarize a few key points for you. We monitor forest composition and litter production on plots to follow how the forest changes over time and in response to drought. Drought is an important factor driving tree mortality in this forest. And major oak mortality after severe drought was caused by interactions between drought stress that weakens the trees and subsequent pathogen infection. Thanks for watching.